Welcome to the class. I am Dr. Shalini Singh, Department of Biotechnology, and topic for today is self-incompatibility in plants. Self-incompatibility refers to the inability of a plant with functional pollen to set seeds when self-pollinated. So it is the failure of pollen from a flower to fertilize the same flower or other flowers of the same plant. And this pollen is functional, but due to self-incompatibility, it fails to fertilize the flowers. Self-incompatibility is a general name for several genetic mechanisms which are present in angiospermic plants, and it prevents self-fertilization and thus encourages outcrossing and allogamy. Thus, it helps us in the production of hybrid seeds. In plants with self-incompatibility, when a pollen grain produced in a plant reaches a stigma of the same plant or another plant with the similar genotype, the process of pollen germination, pollen tube growth, ovule fertilization, and embryo development is halted at one of its stage, and consequently no seeds are produced. Self-incompatibility is one of the most important means to prevent selfing. It helps us to prevent selfing and thereby it promotes the generation of new genotypes in plants. It is considered as one of the causes for the spread and success of the angiosperm on this planet. Classification of self-incompatibility based on the flower morphology. Based on the flower morphology, self-incompatibility may be heteromorphic, which has been further divided into distyly and tristyly, and it may be homomorphic, where it has been divided into two types, sporophytic self-incompatibility, SSI, or gametophytic self-incompatibility, GSI. Based on the genes involved, it may be monoallelic governed by a single gene, diallelic governed by two genes, or polyallelic governed by many genes. Based on the cytology of pollen, it may be binucleate or trinucleate, and based on the expression site of self-incompatibility, may be ovarian self-incompatibility, stylar self-incompatibility, or stigmatic self-incompatibility. General features of self-incompatibility, if you talk about it, it prevents selfing and promotes outbreeding. So, it increases the possibility of new gene combinations. Causes of self-incompatibility may be morphological, physiological, genetical, or biochemical. There is always normal seed set on cross-pollination. It may operate at any stage between pollination and fertilization, and it reduces homozygosity. In plants, self-incompatibility is often inherited by a single gene designated by capital S with different alleles S1, S2, S3, etc. in the species population. These are different types of self-incompatibility which are present in plants. So first is single locus self-incompatibility, which has been divided into gametophytic self-incompatibility GSI or sporophytic self-incompatibility SSI. Two locus gametophytic self-incompatibility may be heteromorphic self-incompatibility is there, cryptic self-incompatibility is there, and late acting self-incompatibility. What is the mechanism of single locus self-incompatibility? It is the best studied mechanism of self-incompatibility. It acts by inhibiting the germination of pollen on stigma. As you know, in the pollination and fertilization process, first step is landing of pollen onto the stigma where it starts germination and pollen tube is produced. So in case of this self-incompatibility mechanism, it inhibits the germination of pollen grain on the stigma. Or if the pollen grain germinates it inhibits the elongation of pollen tube in the style. These mechanisms are based on protein-protein interactions, and the best understood mechanisms are controlled by a single locus which is termed as S, which has many different alleles in the species population. Despite their similar morphological and genetic manifestations, these mechanisms have evolved independently and are based on different cellular components. Therefore, each mechanism has its own unique S genes. The S locus contains two basic protein coding regions. One is expressed in the pistil and the other in the anther or pollen. It is referred to as female determinant and anther is referred to as male determinant. Because of their physical proximity, they are genetically linked and are inherited as a unit. And these units are known as S haplotypes. Translation product of the two regions of the S locus are two proteins which, by interacting with one another, lead to the arrest of pollen germination or pollen tube elongation, whatever is the case in that species which you are undertaking for your studies. And thereby it will generate a self-incompatible response, which will in result prevent fertilization. However, when a female determinant interacts with a male determinant of a different haplotype, no self-incompatibility is created and fertilization ensues. That means if female and male are having same type of genetics, then there will be generation of a self-incompatible response. But if they are belonging to different groups, that means male and female parts of the flower are belonging to the different self-incompatibility groups, there will be normal fertilization. This is a simplistic description of the general mechanism of self-incompatibility, which is more complicated, and in some species the S haplotype contains more than two protein coding regions. So this is sporophytic self-incompatibility. The pollen mother cell, S allele products are synthesized before completion of meiosis. After meiosis, it will be undergoing division. And when it undergoes division, there is a mature pollen which is tricellular. In case of gametophytic, it is pollen mother cells divide and then S allele products are synthesized after completion of meiosis, and these are mature pollen which are bicellular. And these are S1, S2 products. So in case of prophytic self-incompatibility, the pollen is having both of the products S1 and S2, whereas in case of gametophytic self-incompatibility, one pollen is having only one type of protein product, either S1 is present or S2 is present. This is the example of gametophytic self-incompatibility. In gametophytic self-incompatibility, if pollen S allele does not match either stigma allele, pollen will germinate. Suppose the pollen parent genotype is S1, S2. So one pollen will be having S1 protein and the other is having S2 protein. And the stigma genotype is S2, S3. So it is having two type of protein, that is S2 or S3. So these both proteins S2 and S3 are expressed in this stigma. This is S2 protein and this is S3 protein. This is stigma. This is stigmatic surface where these pollens land. So what will happen upon the landing of these pollen grains? This S1 protein, since there is no S1 protein in the stigmatic surface, so this will start germination of pollen tube, and this pollen tube will be elongated and will reach the embryo sac for the fertilization procedure. While this S2 gamete, it will land on the stigmatic surface. It will start germination of pollen tube, but this pollen tube will not grow further as there is S2 protein present in the stigma also. So due to this interaction, the pollen tube growth will be halted and there will not be a compatible reaction with the S2 pollen. So if we see the reaction of this S1, S2 male parent with S2, S3 female parent, it is known as partially compatible reaction. Only S1 pollen tube will be able to fertilize this flower, not S2 pollen will be able to complete the fertilization procedure. In the second case, that is of sporophytic self-incompatibility. Here, if pollen coat S protein is the coat protein do not match either sigma S protein, pollen tube will grow. That means this pollen should not contain any of the protein in its coat also which are present in the stigmatic surface. For example, the pollen parent genotype is S1, S2. So the pollen will be S1 or S2 type. So there are two examples where first female parent is S1, S3 and the second female parent is S3, S4. So this stigma is having two proteins, one is S1 and other is S3. This is having S1 pollen and this is S2 pollen, but they will behave from their coat protein as S1. As we see the genetics, so S1 is dominant over S2. So its coat protein will behave like S1. Its coat protein will also behave like S1. And since S1 protein is present in the stigma, so neither of the pollen will germinate. Whereas in this case, it is behaving like S1. It is also behaving like S1 due to dominance and there is no S1 protein in the stigma surface. Stigma is having only S3 protein and S growing here and will be completing the fertilization procedure. So this reaction of S1, S2 with S1, S3 is incompatible reaction which we call it as self-incompatible reaction, whereas the male S1, S2 when is fertilizing to S3, S4 female plant, then this reaction is known as fully compatible reaction. So in case of gametophytic self-incompatibility, this form of self-incompatibility is more common than sporophytic self-incompatibility, but not so well understood. It occurs in nearly one half of all the families of angiosperms, including the Solanaceae. Solanaceae is a family which has potatoes and tomatoes and tobacco. Petunias, beet, that is Beta vulgaris, buttercups, ranunculus, lilies, roses, and many grasses. The rules in gametophytic self-incompatibility, the S loci are extremely polymorphic. That is, there is an abundance of multiple alleles in the population. Incompatibility is controlled by the single S allele in the haploid pollen grain. Thus, a pollen grain will grow in any pistil that does not contain the same allele. So here this pistil is S1, S2, this is S2, S3, this is S3, S4. Since this pollen is having S1, this is having S2. So none of will grow here since the pistil is having both proteins S1 and S2. This pistil has having protein S2, S3. So this S1 pollen will grow, but this S2 will not grow. So this is fully incompatible reaction, partial compatible reaction, and this pollen is S1, S2 and pistil is S3, S4, so both will grow. So this is a compatible reaction. So self-incompatibility, partial compatibility and fully compatibility reaction. So gametophytic self-incompatibility gives us three types of reactions. This appears to be the mechanism in the petunia. All pollen grains, incompatible as well as compatible, germinate, forming pollen tubes that begin to grow down the style. However, growth of incompatible pollen tubes stops in the style, while compatible tubes go on to fertilize the egg in the ovary. The block within incompatible pollen tube is created by an S-locus encoded ribonuclease, which we call as RNase, which is synthesized within the style, enters the pollen tube, destroys its RNA molecule and halting pollen tube growth. So in brief, this is the whole mechanism of self-incompatibility reaction, that is gametophytic self-incompatibility reaction. This RNA molecule contains a hypervariable region which is encoded by a different allele, which establish each specificity, whether it is S1, S2 or S3. The pollen tube expresses a protein which is designated as SLF, that is S-locus F-box protein, that binds RNAs. SLF also exists in different specificities, S1, S2, S3, etc. So in compatible tubes, the SLF or SCF triggers the degradation of the sRNAs, thus permitting RNAs in the pollen tube to survive and growth to continue. Whereas in case of incompatible reaction, tubes, the interaction, for example, the S1 of SCF and the S1 sRNAs blocks its degradation. So the RNAs of the pollen tube are destroyed and growth is halted. All pollen grains are produced by S1, S2 plant in this reaction. And this is pistil is S1, S2, here pistil is S1, S3 and pistil is S3, S4. So S1, S2, this first one is written as male plant and this second one is female plant. So S1, S2, S1, S2 is fully incompatible reaction, no pollen tube is growing here. S1, S2 with S1, S3 is partially compatible reaction. Only one pollen tube is growing, that is from S2 pollen. S1, S2 with S3, S4 will be a fully compatible reaction since both of the pollen grains are growing here. Second example is of sporophyll. So the control lies in the S locus, which in actually a cluster of three tightly linked loci. First is SLG, that is S locus glycoprotein. It encodes part of a receptor which is present in the cell wall of the stigma. Second is the SRK, that is S receptor kinase, which encodes the other part of the receptor kinase, attached phosphate group to other proteins. SRK, that is S receptor kinase, it is a transmembrane protein which is embedded in the plasma membrane of the stigma cell. And the third one is SCR, that is S-locus cysteine-rich protein, which encodes a soluble ligand for the same receptor, which is secreted by the pollen. So this is the pollen grain, and it is having SCR1, SCR2. This is cell wall of stigma, which is having this SLG. This is plasma membrane, which is holding this SRK. So this SLG encodes part of the receptors, SCR encodes ligand of the same receptor, and this SRK encodes kinase domain of receptor. So this is structure of the S loci in the sporophyte of an S1, S2 plant. Because the plants cannot fertilize themselves, they tend to be heterozygous, that is, carry a pair of different S loci, likewise here shown as S1, S2. However, dozens of different S alleles may be present in the population of the species, that is, the S locus in the species is extremely polymorphic. The difference between the alleles is concentrated in certain hypervariable regions of the receptors. The rules in sporophytic self-incompatibility: pollen will not germinate on the stigma of a flower that contains either of the two alleles, either which are present in the sporophyte parent that produced by the pollen. Like S1, S2 is the pollen parent and S1, S3 is the female parent. So no pollen grain will germinate neither S1 or S2. This holds true even though each pollen grain being haploid contains only one of the alleles. For example, the S2 pollen produced by S1, S2 parent cannot germinate on an S1, S3 stigma. So these pollen grains are produced by S1, S2 parent. So they will be having only one gene, either S1 or S2. And the female parent in first case is S1, S2, so there will be no germination of pollen tube. In second case, if the female parent is S1, S3, but it will behave like S1, it will also behave like S1 because it is influenced by the genotype of the parent plant. So no one will germinate as the pistil is having this S1 protein. And here the pistil is having STS protein, so both will germinate. These are fully incompatible reaction and this is fully compatible reaction. So S1, S2 pollen producing sporophytes synthesize both SCR1 and SCR2 for incorporation, both S1 and S2 pollen grains. If either SCR molecule can bind to either receptor on the pistils, the kinase triggers a series of events that lead to failure of the stigma to support germination of the pollen grain. Among these events is the ubiquitination of proteins targeting them for destruction in proteasomes. In this part is not triggered, that is pollen from an S1, S2 parent on an S3, S4 stigma. The pollen will germinate successfully. So this is the sporophytic self-incompatibility. Here S1, S2 pollen is not germinating on S1, S3 pistil, but S1, S2 pollen is germinating on S3, S4 pistil fully. Next type is two-locus gametophytic self-incompatibility. The grass of family Poidae and perhaps all of the family Poaceae have a gametophytic self-incompatibility system that involves two unlinked loci referred to as S and Z. If the alleles expressed at these two loci in the pollen grain both match the corresponding alleles in the pistil, the pollen grain will be recognized as incompatible. Heteromorphic self-incompatibility. The genes responsible for self-incompatibility in heterostylous flowers are strongly linked to the genes responsible for flower polymorphism. Examples are distyle and tristyle. So distyle. Both stamens and styles are of two types. Stamens may be low and high and styles may be short and long. It is determined by a single gene with two alleles. The flower with short style and high stamens is called as thumb type, and the flower with long style and low stamens is known as pin type. Both thumb and pin flowers differ for 6 characters in addition to stamen and style length. So this is a pin flower and this is a thumb flower. In pin flower, stigma is a long style, situated on a long style, and anthers are short. So it is a pin flower, and in thumb, the stigma is shorter, that means style is shorter, and the pollens are longer in size. So this reaction with thumb flower, they are having two morphology. That's why it is known as heteromorphic. So thumb with thumb will give us incompatible reaction. Pid with pin will give incompatible reaction, but when we cross thumb and pin, it will always give us a result in 1:1 ratio, and that will be a compatible reaction. Tri-styling. In tri-styling, styles and stamens have three different positions. It is determined by two genes S and M, each with two alleles. S give rise to short style, S and M to medium style, and small m to long style. The number of possible genotypes is greater, but a 1:1 ratio exists between individuals of each self-incompatibility type. So this is the long style, this is short style, and this is the medium style. Next is cryptic self-incompatibility. It exists in a limited number of taxa. For example, there are evidences for cryptic self-incompatibility in bladder campion, that is Silene vulgaris of Caryophyllaceae family, Vipers buglos or blue weed, that is Achium vulgare, in and then water willow or swamp loosestrife, Decodon verticillatus. This is bladder campion, Silene vulgaris, water willow or swamp loosestrife, and this is Vipers buglos. They three plants have cryptic self-incompatibility. In this mechanism, the simultaneous presence of cross and self-pollen on the same stigma result in higher seed set from cross pollen relative to self-pollen. However, as opposed to complete or absolute self-incompatibility, in cryptic self-incompatibility, self-pollination without the presence of competing cross-pollen results in successive fertilization and seed set. In this way, reproduction is assured even in the absence of cross-pollination. CSI acts as, at least in some species, at the stage of pollen tube elongation and leads to faster elongation of cross-pollen tubes relative to self-pollen. The cellular and molecular mechanism of cryptic self-incompatibility have not been described. The strength of this response can be defined as the ratio of cross to self ovules formed with equal amounts of cross and self pollen. Last one is late acting self-incompatibility. It is also termed as ovarian self-incompatibility. In this mechanism, self-pollen germinates and reaches the ovules, but no fruit is set. Late-acting self-incompatibility can be pre-zygotic or post-zygotic. That is Narcissus triandrus. Narcissus triandrus, it has the pre-zygotic self-incompatibility. And this is Pathodia campanulata. It has very beautiful flower, and campanulata has a post-zygotic self-incompatibility. The existence of the late-acting self-incompatibility mechanism among different taxa and in general is subject for scientific debate. Criticizers claim that absence of fruit set is due to genetic defects which are the direct result of self-fertilization causing inbreeding depression. And it is a form of the inbreeding depression phenomenon. What is the importance of self-incompatibility in plant breeding? Self-incompatibility effectively prevents self-pollination. That is the most important application of this procedure. As a result, it has a profound effect on plant breeding approaches and objectives. In self-incompatible fruit trees, it is necessary to plant two cross-compatible varieties to ensure fruitfulness. And next point is self-incompatibility may be used in hybrid seed production. For that, two self-incompatible but cross-compatible lines are to be interplotted. Seeds obtained from Both the lines would be hybrid seeds. Self-incompatibility provides a way for hybrid seed production without emasculation and without resorting to genetic or cytoplasmic male strand. Demasculation is removal of anthers from the flower before pollination. Self-incompatibility system permits combining of desirable genes in a single genotype from two or more different sources through natural cross-pollination, which is not possible in some self-compatible plants. In case of pineapple, commercial clones are self-incompatible. As a result, their fruits develop parthenocarpically and these are seedless. Limitations of self-incompatibility. It is very difficult to produce homozygous inbred lines in self-incompatible species. Bud pollination has to be made to maintain the parental line. Self-incompatibility is affected by environmental factors such as temperature and humidity. Incompatibility is reduced or broken down at high temperature and humidity. There is a limited use of self-incompatibility due to problems associated with the maintenance of inbred lines through hand pollination as it is tedious and costly. References you can study from any book of your choice. But Principle of Plant Breeding by B.D. Singh has very fairly explained this topic. Thank you so much for listening carefully. If you have any question, you can always get back to me. Thank you so much.